got three phases. Starts with the reduction, carbon fixation or oxygenation, proceeds to the reduction, then eventually regeneration. And you know that in between the reduction and regeneration phase, a small amount of glucose or sugar precursor exits the cycle, while the most of it continue the cycle to regenerate and do the cycle all over again. And this is very important to ensure the stability of 5-carbon sugar RUBP to be always in the stroma of chloroplast. Because if the cycle is not a cycle, just a simple biochemical reaction, linear, RUBP is going to be all used up and the sugar precursor production will be halted completely. And plants do not want that to happen. Okay, sugar needs to be created at all time. Not sugar precursor, because this precursor not only meant to make glucose. Throw away that understanding from you now. Sugar precursor can be turned into storage molecule, such as what? Starch. Can be turned into signaling molecule, such as sucrose. Can also be turned, in fact, to some kind of other molecule, such as the B vitamins, right? So vitamins, some certain B vitamins actually use this sugar precursor as well to do the manufacturing. That's right. Now we proceed to the second part of it, which is the photorespiration, and this is actually happens because. So by the way, Calvin cycle, the other name for it is C3 cycle. There's so many. Pentose phosphate cycle, C3 cycle, Calvin cycle, dark cycle, so many other names, synonyms. However, the star enzyme involved in Calvin cycle, the Rubisco, ribulose bisphosphate carboxylase oxygenase, is an ancient enzyme. Therefore, it has the issue not to be specific enough to catalyze single sole reaction, which is the carbon reaction up here, right? If it proceeds with this carboxylation reaction, then that's fine. You get your phosphoglycerate that can be reduced to become your sugar precursor. However, the issue with Rubisco is it can add up oxygen to the RUBP and proceed with, many like to call it a wasteful process. I would like to call it metabolically expensive process called photorespiration. When it proceeds this way, it will create two molecules. The first molecule is the regular phosphoglycerate to proceed with the regular Calvin cycle. The problem is here. It creates the 2-carbon product, not 3-carbon, 2-carbon product called 2-phosphoglycolate. This thing actually is a bit toxic to the plant. So what the plant does? The plant needs to eliminate this or need to turn this back so that I can reuse this carbon for the sake of making sugar precursor again. Because this thing, another problem is it cannot be converted directly from here, jump to here. Plants do not have the enzyme. Unless some kind of mutation happens, then we do not have this photorespiratory issue. Right, so let's see. Just need to turn this. We need to make this big actually. Macam tu. Right, okay. Fair warning. This is going to be a bit overwhelming. Why take so much oxygen in you? Open your brain so big, so wide, so you can somewhat catch the cycle here. So photorespiration is a type of respiration, meaning that it consumes ATP. It breaks down something to get something, but the trouble with photorespiration is it's a lengthy and expensive process. To start with, photorespiration involves three organelles: chloroplast, from where it started originally, then peroxisome, then the third one is mitochondria. Okay, mitochondria, that's the singular, mitochondria, that's the plural of it. So these three organelles, without the oxygenation reaction done by the Rubisco, these two really are of the question. Chloroplast alone is enough to make the sugar precursor. Now, since the plants have produced this toxic two-carbon product, phosphoglycolate, these need to be turned into something useful again. So the whole idea is from 2-phosphoglycolate, how to get back to 3-phosphoglycerate. You see, it's just from here to here. But there is no enzyme to do this. So in order to do that, plant has to go all this path, go back, go down to the peroxisome, mitochondrion, coming back to peroxisome, coming back to chloroplast. It's such a long process. So you are equipped now with the enlarged version of this so that you can study on later. Right. So for the second page of this is the summary of the reaction that's going to happen. But let's be realistic. Yeah.
Why there is a question mark?
Oh, I'll come to that in a bit. So you see these light purple boxes here. These are called transporters. Transporters meaning that it's like a portal or doors from which molecules can exit or enter. Okay. 2 years ago, when I taught this photorespiration, people have not confirmed to find the name of this transporter. So this transporter actually, okay, give you the name. So it's PGGG1 and BASS. So P stands for plastidial um, glycolate glycerate, number one, and BAS, what is that? This is, by the way, this is transporter, okay? This is called transporter. Transporter. This BASS6 is called symporter, meaning that it one door used by many people, the sim. BAS is stand for bile acid, sodium symporter. 6. Whenever you see this question mark, meaning that people have not found the name or the protein involved to make the. So this is the research opportunity actually to study this. I tell you, it's not easy. It's not easy. In my thesis, there is a technique that can be used to detect this. If you ever read my thesis in chapter 5, I describe there is a technique to detect this. Right. Okay. Coming back to this cycle. I just want to be realistic about this. So I don't like this marker. It's a bit. Marker pun. Tu pun nak habis dakwat dah. Bad day not to have markers. I want to write a lot. Anyway, the realistic bit that I'm talking about is you to cover this in one lecture. You need to find a happy, peaceful place to read it and to absorb. But what I can assist with you here is to create the story so that you can see that it starts with something, then it come back to something, right? So let's start somewhere here. So I think it's not going to be in circle, it's going to be in L. It's not that big, right? Okay, so let's start. What? This thing need to, well, dia punya like masuk atas. Ada marker pun tak baca kat bawah. Baca tu okey. Pinjam. Pinjam. Okay, so we start in chloroplast. Yep. This is good. So, from last week, you learned that RUBP, which is the 5-carbon sugar, will react with Rubisco that has been activated. Remember, this needs to be activated from your last week lesson. So, in activated form, it called ECM. Is it? You need to recall back your the Rubisco has been carbamylated. Okay, it has been carbamylated with CO2. Remember, okay, the CO2 needs to activate Rubisco. It's not the same as Rubisco react with CO2. This is just a free CO2 attached to Rubisco and then stabilized with magnesium to form the activated form of Rubisco. Okay, then this will plus with H2O. Oh no, we are talking about photorespiration, right? Photorespiration just straight to oxygen, and then this will produce. Okay, we are in chloroplast. This will produce one molecule of phosphoglycerate. This is fine, nothing problem with it. Plus phosphoglycolate. Yeah, 3 carbon. With this proceed with the regular Calvin cycle. Now the issue is phosphoglycolate, right? Now the problem begins. So this phosphoglycolate, I think I just put it 2 pg, okay, to simplify it. It needs to become phosphoglycerate. This is the idea. However, there is no straight conversion, so it needs to begin the journey now to convert itself. How does it happen? It needs to be phosphorylated first. So it will undergo phosphorylation, dephosphorylation actually. Dephosphorylate. Now it will become a glycolate plus ADP plus PI. Okay, the name of the enzyme is actually on this page. So what's the name of the enzyme? It should be phosphatase. Phosphatase is to remove phosphate. Dephosphorylation. The enzyme here is phosphatase. Glycolate phosphatase. Is it glycolate phosphatase? Phosphoglycolate phosphatase. Look at number 2. Okay, meaning that it now become glycolate after phosphate has been removed. Now, this glycolate can now exit the transporter. You got glycolate, right? This glycolate do not stay forever in chloroplast. It will now leave the chloroplast and then through this transporter and symporter, you got two here, two doors here. It must be very toxic, otherwise why plant use two doors to let it go out? It go out and get into the peroxisome. So now you are in peroxisome. Oh, boleh sebenarnya tekan kuat sikit. Peroxisome. Okay. So what happens to glycolate in peroxisome? So glycolate in peroxisome will be oxidized. Okay. So when it oxidized, meaning that oxygen is what? Added or removed? Removed. Look at this. Oxygen is added to it. Okay. So the enzyme responsible for this should be glycolate oxidase. Is that correct? Is it? Yes or no? Oxidase. To yield glycosylate. Okay. So plus H2O2. This is peroxide. That's why the name is peroxisome, because it produces peroxide. So you get your glycolate reacts with oxygen. Now you get a new compound called glycosylate plus peroxide. Okay. This glycosylate will proceed with another reaction. However, just a side reaction, this H2O2 is very, very damaging. Right? It is so damaging because you can use it to bleach. So plants will break down H2O2 on the sideline using the enzyme catalase. So catalase, it breaks down. Now you get H2O plus oxygen. 2 times of breaking down, you will get a molecule of oxygen, O2. Okay, and the reaction here, in here, will be number 4. This is the main reaction. Glycolate converted to glycosylate. Glycosylate proceed with the next step. However, H2O2 peroxide is very damaging. So this hydrogen peroxide on the sideline will be neutralized. You can call it neutralized. Will be neutralized by the action of catalase. Catalase means it breaks down. It breaks down as a water and oxygen. 2 times breakdown, you got a molecule O2 of oxygen. Okay. Now coming back to glycosylate. So what happens to glycosylate now? So this glycosylate, it will now react. Remember, you are still in peroxisome. Okay. Glycosylate now reacts with an amino acid called glutamate. Okay, glutamate, and this is a special process because it uses one amino acid to make another amino acid. So in this case here, glycosylate here, reacts with glutamate in order to produce glycine plus 2-oxoglutarate. Okay, and the enzyme responsible for this conversion to get glycine and 2-oxoglutarate, this is called is it glutamate? Glutamate. No, not glutamate. Or glyosylate. Should be glyosylate first. Glyosylate column aminotransferase. You see this enzyme aminotransferase, meaning that this process here in the book you will say is called transamination. It uses glutamate and then it reacts with glyosylate to get a new amino acid. Glutamate is an amino acid. Okay? Glutamate is an amino acid. But this enzyme do the reshuffling and rearranging to get a glycine. What about this 2-oxoglutarate? These actually maybe when you learn your biochemistry, do you learn Krebs cycle? Krebs cycle has a molecule in there called alpha-ketoglutarate. So this is the synonym as alpha-ketoglutarate. It is an intermediate in the Krebs cycle or citric acid cycle, right? What's the function of this thing? This thing actually you will see in the cycle involved over and over again whenever there is an involvement to create an amino acid from other amino acids. Okay, meaning that this newly synthesized amino acid not from any precursor, it's actually from other amino acid gets converted. When conversion happen through the process of transamination, this is needed. Okay, to prove you to that point, you can see that 2-oxoglutarate is being used again in reaction number 8. When you want to convert amino acid serine to hydroxypyruvate, you use that again, right? And number 12, you use that again to convert glutamine back into glutamate. They're changing from one form to another. Okay, right. So oxoglutarate, we are not talking about it because that is the reactants in many other things. You see, oxoglutarate can go here, can jump here, can go to chloroplast. Now we want to proceed here. Now what happened to glycine here? So this glycine, it will proceed to the third organelle. What is that? Mitochondria or mitochondrion. Glycine. So in the mitochondrion, this glycine can be converted to serine, another amino acid, and in the process, it releases a number of things. It releases NADH, it releases CO2, it releases ammonium ion, NH4+. You see, you start to get back your CO2 here. This is the first step, you start to get back CO2. Just now, you have lost your CO2 in the form of 2-carbon phosphoglycolate. Plants want to release back this. I want CO2. Stop making unnecessary compound. So this is the first step. CO2 is retrieved back. And based on this table, that would be reaction number 6. Is that correct? Yep. Number 6. It might look a bit complicated in there because glycine to serine is not a straight process. Okay, what happens is, I know it looks a bit complicated there. Let's put it this way. Glycine needs to react with NAD, NAD plus. Okay, then it reacts with GDC. This is actually an enzyme. Glycine decarboxylase complex. When you see it in this form, meaning that this thing is not activated yet. Okay, it's not activated yet. When this thing is activated, when this thing happen, you will get, I'm talking about reaction number 6, okay? You will get your GDC TH. Oh, that's a long name. Tetrahydrofolate. And then CH2 plus NADH plus CO2 plus NH4 plus. Do you see any syringe yet? No, there's no syringe because this is the key molecule here. How do you pronounce this? Such a long mouthful thing. Activated methylene tetrahydrofolate. Okay. Originally, it's in this form, just GDC here. Now, once it becomes activated, it becomes this form. And guess what? This thing, now we move to reaction number 7, okay? I think number 7, should be number 7. So this GDCTHFCH2 react again with glycine. You see, just now it would react with glycine to activate itself. Now it reacts again with glycine to get what? Serine. Number 7 here. Oh, there's a water here. So you will get serine. How does this happen? Remember, whenever there is a water involved, the name of the enzyme is not going to be just simply decarboxylase synthase. It will be a weird name, something like this. So this is serine, right? So it should be the enzyme involved for this reaction is called serine hydroxy methyl transferase. Ooh, how I know? You want to create serine. There is a water involved. And then this thing got a methyl. You see? CH2 is methyl. You want to transfer this methyl group to glycine. When methyl is transferred to glycine and then gets hydrated, you will get serine. Right? You will get serine. And guess what? The moment it has donated methyl to glycine, it becomes what? It becomes deactivated again. It becomes back the GDC. Then this thing can be used again here. Right? Okay? Okay? Right. Okay, can follow now? Where are we now in the organelle? Which organelle are we now? Mitochondria. Still mitochondria. We're still in mitochondria. So now we are interested with serine. I'm losing a board now. Maybe I can pull this up. This thing should be good enough, right? To see the notes. Okay. So I'll just rub this, okay? Because you already know what GDC stands for. Need all the space. Oops. Did I double? No. Okay. Now, tak ada awak lah. Semua warna kena guna. Oh, this is smooth. Now, this serine here needs to exit the mitochondria. Now go where? Go back to peroxisome. Now we are in the peroxisome. Okay? What happens now? So in the peroxisome, serine can again react with what? Glutamate to produce what? Sorry, not glutamate. Sorry, not glutamate. It wants to produce glutamate. It reacts with this thing because it needs to change itself again. Remember, amino acid wants to change itself again. You always need this. 2-oxoglutarate. Okay, and the name of the enzyme here should be glutamate oxoglutarate amino transferase. To get what? To get glutamate plus, this is critical, hydroxypyruvate. Okay, I need to make sure it's all the way out. Okay. So what happens to hydroxypyruvate is, hydroxypyruvate, don't worry about the glutamate because this glutamate will be recycled to be used again here. React with glycerate because this is peroxisome, remember? We saw earlier glutamate is used in a reaction. Reaction where? With glycerate. Glycerate react with glutamate in order to get glycine, right? So we're not going to talk about the glutamate. We're going to proceed straight to the hydroxypyruvate. This hydroxypyruvate it can be reduced with the help of hydroxypyruvate reductase plus NADH plus H to get what? You will start to get back your glycerate. You see? You see the magic there? So this thing has become plus NAD plus. So here's the magic happen. You start to get back your glycerate. Remember? Isn't it the very thing that will be the basic of Calvin cycle? However, for Calvin cycle to happen, the first molecule is phosphoglycerate. This newly created glycerate will be phosphorylated. So glycerate react with glycerate kinase plus ATP. Now you will get your phosphoglycerate. And this phosphoglycerate is actually this thing that proceed with the regular Calvin cycle. And Oh, I forgot to tell you, this glycerate now has come back to chloroplast. This is actually in the chloroplast. Hydroxypyruvate that has been reduced to form glycerate and NAD. The glycerate will move back to chloroplast, and then it will be phosphorylated by a kinase, glycerate kinase, to become 3-phosphoglycerate, right? And you just save the carbon from missing using this one very long process, okay? However, this only talk about the carbon cycle of it, right? So this is why photorespiration, the other name of it is C2 cycle. C2 cycle. C2 or C2 metabolism. How many C have you learned? C3, which is Calvin cycle. C2, photorespiration. Is there any other C? This here. No, this not C4 later. C1 cycle. From GDC, activate, and then come back to GDC. You see? Isn't it a cycle? This is C1. Methylene is 1 carbon. So this is actually C1 metabolism. Not involving to make sugar, but involving methylene going back and forth between GDC in order to activate it. If it is not activated, glycine cannot be converted to serine. If serine is not present, phosphoglycerate cannot be retrieved back. It's as simple as that. Simple, right? Okay, all right. Okay, just after the one. Don't worry about reaction number 11 and 12. That is actually more on the nitrogen cycle. Okay, I just quickly mentioned that. For reaction 11, you will see that glutamate, which is a lot here, you got many glutamates here now, can react with ammonium ion. Where did you get ammonium? When glycine is converted to serine. You see, you got ammonium ion. This ammonium ion is actually at loss because it is not attached to anybody. So plants need to get it back. To get it back, it reacts with glutamate with the help of ATP to form a new amino acid called glutamate. Right? Okay? Yeah. And because the needs of the glutamate is rather high, especially in the photorespiration, the glutamine, when needed, it can revert back to glutamate by the help of, remember, from one amino acid to another amino acid, you need 2-oxoglutarate. So 2-oxoglutarate reacts with glutamine plus, what is this? FDRAD. You found this over and over again. Last week you found it again. Pyridoxine in the reduced form. But you wonder, how come pyridoxine is here? Aren't we talking about Calvin cycle now? Shouldn't Calvin's pyridoxine in the thylakoid? If you recall back, pyridoxine is actually in the stroma lamellae. Stroma lamellae is not bound to the membrane of thylakoid. It's loose. It can go back and forth wherever it wants, right? Yeah, you react with this, then you got your hydrogen, then you get your glutamate some more, plus pyridoxine in the form of oxidized. Okay, so in this way, you get actually 3 things recycled. Number 1, you got carbon, because at first you lost and then you get it back. This is the carbon cycle. Then you got your nitrogen cycle. NH4 is released to the stroma, but it reacts with glutamate in order to get glutamate. Now it has been retrieved. Where else? You get oxygen cycle, number 3, in here. Oxygen is used to react with glycolate. Glycolate is the second thing happens after the oxygenation of Rubisco. So glycolate react with oxygen to produce peroxide. However, you don't want this. You want oxygen back, okay? Because this is a living thing. You don't breathe using peroxide. You breathe using oxygen. And also you need the water. So this peroxide undergo catalytic reaction with the help of enzyme catalase to yield back water and oxygen. And this is the oxygen cycle. So you got 3 cycles now. So plants in the inefficiency here and somewhere, it still managed to obtain the aims of metabolic stability somewhere, right? So after you've learned this about photorespiration, we come back to the original statement of many people like to call photorespiration is a wasteful process. Do you think it's a wasteful process? Is it wasteful? In to our interest, because we always want sugar precursor, it is a wasteful process because it uses ATP, it uses ADP just to go all the way to get this back. Because it cannot convert from phosphoglycolate to phosphoglycerate. However, for the plants, this is a scavenging mechanism to retrieve back the carbon so that the carbon is not lost. To make sure that if something get released in the environment, such as ammonia shouldn't be in the free form, okay? Suddenly, it's randomly happening. This need to be in somebody, usually in amino acid, okay? So this is actually a protective mechanism to make sure that at least the leaf do not undergo photobleaching. You know photobleaching? The leaf is under intense light for quite some time. Then it starts not to photosynthesizing. Then eventually it will start to get pale in color. Photobleaching, right? Or photoinhibition. Yeah. So it's a protective mechanism actually. But of course, in order to protect, you need to use energy, you need to be inefficient about it. It's pretty much like you. You like to go to the beach and you don't put any sunscreen. Eventually you got skin cancer. Now you have to pay to the doctor and dermatologist to treat your cancer. It's very expensive. But you need it in order to stay alive. You need to spend the money in order to stay alive. You need to spend money to pay for your tuition fees, your registration fees. Kan bodoh pula. You don't go to university, right? Something needs to be at loss in order to gain something bigger. Right. So that actually is the summary of this whole reaction. This thing, you cannot press that stop button. It will restart this thing for some reason. Okay. So this is the overall reaction that I have just mentioned. Something for you to read. This thing you need to relax, soothing. Baca. Okay. Okay. Should I use back the screen? Anyway. Okay. This thing actually I have explained to you why glycine conversion to serine is not one process. So on this page, I have explained you to it here. How glycine needs to reactivate the GDC-THF. Oh, I missed it. The THF here. Tetrahydrofolic. You know folic acid? You know, if you go to donate blood, if you're pregnant, doctor give you folic acid, right? Not because the chloroplast is pregnant. folic acid is needed actually to activate this GDC. Right. So if you think about it, nutrients needed are the same, but for different reasons. You need the folic acid so that your baby is not weird, and also you get some more blood. Plants need folic acid so that glycine can be converted to serine. Yeah, if serine is not happening, you're not going to get back your carbon, right? Okay, so in this whole process, there is a change of NAD to NADH, even though this is not a regular mechanism of respiration in mitochondria. You get it anyway, because later when you learn respiration, you get this conversion from NAD to NADH. NAD is called the pyridine nucleotide. You know, pyridine is a form of structure. Nucleotide is anything that has the nitrogenous base, phosphate backbone, such as adenine. You know, all the A, T, C, G in the DNA. So those things are called nucleotide. This is not DNA, but it has the nucleotide here. Yeah. So NAD, the other name of it, I know all this while people call it reducing molecule. You can call it now as well pyridine nucleotide. Is it? Yeah, it says here. Pyridine nucleotide. There's only two. It's either pyridine or pyrimidine, if you remember your biochem lesson. Right. So this is the overall of it. You got your cycle, you got your reactions, okay? Right. So what is expected for you to know? You need to know more or less this thing. What happens in each organelle, okay? Don't tell me you get serine in peroxisome. Masa tu kau kena bomb. Mana ada serine tu dalam mitochondria je. Don't tell me you get catalase in mitochondria. That is wrong. Catalase to break down hydrogen peroxide. It should be in peroxisome, not mitochondria. Right. Right. And this is, I have also mentioned this about the overall cycle of carbon, nitrogen, and oxygen. So for the carbon, this is the upper bit of it. So you got RUBP, you got phosphoglycolate, and then go through the peroxisome, mitochondria, then you get your glycerate back. Carbon cycle. Nitrogen cycle, so you get your nitrogen, NH4. NH4 can go back to chloroplast to become a new amino acid, which is glutamate. Ammonium plus glutamate, you can get glutamine, right? And then oxygen cycle, which happens within the peroxisome. There is no oxygen cycle in mitochondria. There is no oxygen cycle in the chloroplast. Don't get it wrong. Look at this diagram. It's within. What is this exactly? This thing. Glycolate reacts with oxygen to produce glyoxylate plus peroxide. Peroxide breaks down to produce water and oxygen. Then this can use again. See oxygen? It can react back with glycolate. Over and over and over and over again. Nature full of cycle. Right. You got 4 seasons. You got annual plant. Right. And so on. Ooh, what's this? What time now? I'll just spend five minutes here. So here what's going to happen today. After this, we can go to ladang for a bit because we need to spray in the morning. We'll spray your treatment, T2 and T3. However, this has been prepared for you earlier, 2 days back. So in the afternoon, you learn how to prepare this treatment solution. Plus to finish 3 slides, 4 slides more. All right? Okay? Just like last week. Right? Because I don't want to bomb you with this. Have you learned, do you know Michaelis Menten? Did you learn this? Michaelis Menten. You learn about enzyme, right? When you learn about enzyme, did you learn about Michaelis Menten? If you do not know Michaelis Menten, I'm very worried because I need to explain it. Did you learn in biochem? How come you learn enzyme tapi tak belajar Michaelis Menten? Okay, let's put it this way. Enzyme is a protein. Let's start with that. It's a protein. It is actually not part of the reaction. It just catalyzes the reaction, or it just boosts up the reaction so that the reaction can happen faster and the reaction can happen specifically, meaning that there's no room for error. That's the function of enzyme. Okay, and also to lower the activation energy. For example, without enzyme, the reaction can happen, but it takes a very long time. So the enzyme comes in, the energy needed is less to do the reaction, so the reaction happens at much efficient rate, right? So in any chemical reactions, you got your reactants, your substrate. Okay, so for any enzyme to work, it needs some kind of affinity or inclination to bind with its substrate, like in here. So you got enzyme A, and the enzyme is the Pac-Man here. The substrate is the small circle up here, right? So the enzyme need to attach to the substrate. However, this attachment of enzyme and the substrate is governed by other external factors such as temperature, pH, as well as substrate concentration. Okay, so that's why you can see we have two types of enzyme here, enzyme A and enzyme B. Enzyme A, you have 1, 2, 3, 4 enzyme, 1, 2, 3, 4, 5, 6 substrate, only two out of 6. How much percentage is that? 2 out of 6. 30%? 30%? About that. Only 30% of the attachment is happening. Compared to enzyme B, the substrate is less now. There's only 4 substrate. But from these 4 substrate that is present, 2 already gets eaten by enzyme. So the affinity for this is 50%. So you can say that enzyme A and enzyme B is more active, more sensitive, catalytically more vigorous compared to enzyme A. So what exactly is Michaelis Menten? It's a name of a person actually. So Michaelis Menten is actually this km, not kilobiter. It's actually the subscript m. It's actually the concentration of substrate at which half of the activity of the enzyme are occupied, right? So if the rate of reaction is here, you are finding the halfway of it. At this halfway, all of these enzymes are occupied by the substrate. So that value here is the Km here, the Km value. So therefore, the smaller the Km, what will happen? High affinity, high attachment. Compared to the higher Km or large Km, low affinity of enzyme for substrate because of high concentration, but you still need more of it to get attached. Okay, so that's the basic of it. So what to do with photosynthesis? We learn this because of Rubisco. Rubisco, remember, it has dual attachment, oxygen and also the CO2. So the problem is, in order for Rubisco to catalyze carboxylation reaction, the amount of substrate, in this case CO2, must be high enough to outcompete oxygen. You can't stop oxygen from being present. Oxygen is always there. However, CO2 just need to be just a bit more so that oxygen is displaced and Rubisco only see CO2. Right. It puts dominance to it. Right. And this is the problem now because CO2 in the form of liquid. Okay. CO2 presence surrounding Rubisco is affected by a number of things: temperature, pH, and also the opening of stomata. The more the stomata open, the more CO2 can get in. But if it's very hot, stomata going to close, right? When stomata close, less CO2 get in, so less CO2 will be surrounding of Rubisco. Yeah. So in order for the plant to make food, to do the carboxylation reaction, CO2 need to say to oxygen blah 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 blah, so that it can the one gets attached to the Rubisco. Okay, so this is something for you to read. Okay, I'm not going to go through this. This is actually an extra note for you. VC max is actually the rate of catalytic reaction. The specificity, CO is cofactor, right? Whenever you see this square bracket, that means the partial pressure of it. The pressure of, because this is gas that has been dissolved in liquid, right? Yeah. So it's all about understanding how oxygenase and carboxylase reaction is actually happening at the same time throughout the day. However, during the peak time, we want the carboxylase reaction to happen more so that you get more sugar. Yeah. So this is when you learn that the plants employ so many mechanisms to ensure that this Rubisco keep on doing carboxylation reaction. The pH issue. The solubility of CO2 is decreasing with higher pH. Okay, higher pH. So when the pH is slightly lower, more CO2 can dissolve in the chloroplast. So more is available for the Rubisco to attach to it. And how this is achieved? You have to thank magnesium for it. And also you have to thank the light reaction for it. Because when light reaction happens, the Z scheme, hydrogen from the stroma is pumped into the thalacrot, right? So you have less hydrogen in the stroma. When less hydrogen in the stroma, that brings down pH. Oh, that brings up pH. It becomes more alkaline because less hydrogen. When it becomes more alkaline, more CO2 can dissolve and be present surrounding Rubisco. Yeah, and this CO2, the moment it has attached to Rubisco, don't get too happy too soon. It can be immediately get displaced. Okay, just like the carbamylation, Rubisco need to be activated, right, by CO2. This can also be displaced easily. That's when the magnesium comes in. Magnesium 2 function to further lower the to further alkaline a bit the situation, and also to stabilize the carbamylation, and also to stabilize the carboxylation reaction of CO2. Meaning that you can think of magnesium like a glue. That glue that ensure CO2 stay where it needs to be on Rubisco and also RUBP. Yeah. So and another is temperature, of course. Okay. The higher the temperature, the less CO2 is dissolved in the solution. Right. And this is not only for chloroplasts, even if you do in experiment in the lab. Right. Okay, so that's about the Rubisco function. So we're going to stop here. So we'll continue a bit more, just 3 more slides. And I'll see you in the afternoon. Okay? But after this, can we go to the farm? Just to spray. You learn how to make the spray in the afternoon. Right. Okay. Any question? Any regret? Too late. Too late. Too late. Just a quick advice. This thing, usually when you learn in the lecture, it's all about you learn a fact. How you learn a fact. Apple is red. Earth is sphere. Just that. Most of the lesson that you learn. Once in a while, when you are a bit unfortunate, you come across this kind of lesson. It's not a simple fact. It's actually a sequence of story. Kind of like Cinderella, very unfortunate, then got bullied by the stepsisters, then need to clean the whole house, then got stressed, then escape the house, go to the palace to do the dancing with the prince, and then come the midnight, got panic, then left the show, then come back to home. There's a sequence there. Right, so the story, biochemistry, the pathway cycle story, kind of like Cinderella story. One thing needs to happen first before other thing. You cannot suddenly swap. Instead of Cinderella go to the dancing with the prince, now the stepmother comes in. What? I'm wearing gown too. I'm wearing glass slippers too. Oh, I can do the dancing. Yeah, tapi kau kan Cinderella. Right, it doesn't matter. So the story is incorrect. Right. The whole publisher need to burn the books and need to reprint again. Right. So think of it this way. It trains your brain to know this sequence. How just one thing about this cycle, it's not a linear. Cinderella from beginning until the end, right? Happily ever after. However, for this kind of story, you start with something, you come back with something again. It's a circle story. Do you know any story that start with something, come back with something? I'm thinking. Macam tak ada. Macam tak ada. Or maybe lah kot. Maybe like Adams and Eve started in heaven, then got kicked out of the heaven, then come back to heaven at the end. Kind of like that. But there's a very long eons of years of story. Alright, okay. Dah. So, see you later, okay? Ambil marker balik. Terima kasih. Warna hitam tu macam dah.
Ada, aku dah ambil.